Okay, so next up is Magnus Haughey on the use of spatial genomic data to reconstruct somatic evolution.
Hey, thank you very much. Okay, so my name is Magnus Haughey. I'm a postdoc working with Benjamin Werner at Barts Cancer Institute in London. And so I'm going to talk today, kind of briefly summarize a couple of pieces of work in the direction of using spatial agent-based modeling and combining that with spatial genomic data to understand tissue dynamics. So before I go on, I'd just like to thank my collaborators. So first of all, my postdoc supervisor, Benjamin Werner and his lab, and also colleagues at QMUL Maths Department, Stuart MacDonald, Winnie Huang, and Trevor Graham, and members of their labs as well as Imran Noorani from UCL. So just very quick kind of motivation for using spatially explicit models. What sort of benefits can we obtain from using spatial models? So if you, on the left, I've sort of listed a few sort of generic things that you might want to define when you are using a spatial model, such as defining the lattice and cell neighborhoods. All of the modeling I'll talk about in this talk is lattice-based modeling. And also defining properties of the cells and the tissue that you're simulating. But then crucially, what you then obtain, or what you can benefit from using these models, is you get an understanding of spatial genetic patterns. And importantly, a kind of understanding between how tissue architecture and evolution affect each other and this interaction. And you can also then tailor your models to, more specifically, to different tissue types and sampling strategies. So I'm just going to kind of talk about a few pieces of work, really two pieces of work, and then motivate a kind of where I'm working on now in this direction. First, the first piece of work is where we use spatial agent-based models to understand the dynamics of cell regeneration in healthy human liver. And then next I'll move to cancer, where we set out a framework to kind of quantify the geometrical shape of tumor subpopulations to understand their underlying dynamics based on their geometry. And then last, I'll talk just a little bit about work on understanding spatial patterns of extracellular DNA. So let's start with the human liver. So as many of you will know, the liver has this really incredible property of being able to regenerate lost liver cells. But the mechanisms by which this is achieved, especially in normal human liver, are not particularly well understood. So the liver has this sort of this structure shown here, where it's made up of these repeating hexagonal units that are called lobules. And these are kind of made up of a branch of the central hepatic vein in the center, surrounded by what are called portal triads. And these are three structures: the terminal branch of the portal vein, the hepatic artery, and the bile ducts. And then the liver itself is predominantly made up of hepatocytes. It's the main type of liver cell. And as I said, the mechanisms by which new hepatocytes are generated, especially under homeostatic conditions, are not particularly well known. And an important question is about, do hepatocyte progenitor cells exist? And if so, what is their location and identity within the lobule? And so we set out to understand this question. And having a better understanding of this will then give us a better kind of framework to understand developments of diseases and such. So we set out to explore this by exploring spatial patterns of DNA mutations, mitochondrial DNA mutations in healthy human liver samples. And we did this by identifying clonal expansions of liver cells, which are shown in blue here, and these in human samples. And these tended to sort of emanate from the portal triads, which you can see highlighted at the top of the image, and sort of extended towards the central vein. And so we wanted to understand by taking multiple samples in a line connecting these two regions, and then exploring the spatial patterns of mutations. Do these spatial patterns tell us anything about whether progenitor cell populations exist and anything about their dynamics. And so our experiment, which was performed by my collaborators, it's summarized here. So after highlighting or identifying these clonal expansions of cells within the liver, we then we sampled multiple separate sections of tissue along the portal triad to the central vein at varying distances, and then looked at spatial patterns of the mutations. We looked at patterns of mutational burden across this region. And then on the far right, the plot shows how we sort of then compared adjacent sections and looked at how many mutations were unique or versus shared between these two pairs of sections. We then simulated this kind of underlying process with a spatial agent-based model, two-dimensional model, where we simulated homeostasis in the liver through rounds of cell killing and regeneration coupled to mitochondrial mutation. And we did so with a sort of synthetic kind of fixed PT portal triad progenitor cell population in the center of the system. And what we wanted to understand is what, by varying properties of these progenitor cells, such as the relative rate of division compared to normal hepatocytes, which populated the rest of the system, how does this affect the spatial patterns when we then spatially sample our data in a similar way to experiments and look at spatial patterns of mutations? And this modeling led to these two kind of main conclusions. So the first on the left is that we found evidence for the existence of dedicated progenitor cell populations that are active during homeostasis in the liver, and that these replicate faster than mature liver cells. And this was sort of summarized by this plot here, where we, for each of our human samples, we used approximate Bayesian computation to infer the relative rate of replication of these progenitor cells compared to mature cells. And we consistently found evidence for a faster rate of replication. And then on the right, we used actually a slightly simpler one-dimensional model of the same process. And this allowed us to explore the temporal dynamics of the expansions by comparing mutation repertoires between adjacent sections. And we found evidence that these expansions occur slowly and then quiesce. And we were addressing a kind of long-standing debate in the literature about quiescence versus a kind of continuous conveyor belt like dynamics. We found evidence here for quiescence of these clonal expansions of cells. So then next, I'm just going to talk about a second piece of work where we explored how geometrical shape of tumor subclones can tell us about their dynamics. So in our lab, we were interested in colorectal cancer. And so when we obtained this spatial data, we were really interested to understand what information is contained within this data itself about the underlying dynamics. So this method was published a few years ago. It's called BaseScope. And this is an assay that allows us to stain solid pieces of tumor tissue and probe a specific genetic locus and highlight with high spatial resolution areas of tumor cells that are mutated or wild type at this particular locus. And so you can see these very kind of interesting complex shapes that this data, this method produces. So we wanted to understand what information is contained within the geometry of these wild type and mutant populations, how they mix together, how they're segregated in space, and what metric can we use to actually extract this information. So we explored the use of first passage time as a metric to quantify these complex shapes and extract dynamic information from that. First passage time is it's denoted tau up here, and it's sort of an expression of the mean, the average length of a random walk between two types of nodes. So in here, you can see an example of a yellow to yellow time and then a red to yellow first passage time at the bottom. So we simulated random walks on the pixels of these images, like you see on the left, and quantified first passage time, therefore quantifying these kind of complex shapes of these tumor subpopulations. But to develop this framework, we first applied the first passage time measurement to simulated patterns of wild type and mutated populations that we generated with a similar spatial agent-based model, where we varied. So we simulated a co-expansion of wild type and mutated populations, and we varied the strength of subclonal selection, and also the time at which the mutant population appeared in the system, as well as also the kind of strength of cell pushing on the lattice. And what we found is that when we simulate many different tumors like this for different underlying subclonal dynamics, and then analyze each of these in the phase space of first passage time, so we're quantifying the shape of the mutant patterns, we find that we can actually kind of extract some of the information about the underlying dynamics used to generate these patterns purely by analyzing the shape of the populations that these dynamics give rise to. We then applied this method then to experimental and colorectal cancer samples. And so you see the results on the left here, again, in the same phase space of first passage times. And on the right, you see a few examples of our experimental patterns. And then on the right of those, the kind of best fit simulated pattern ascertained using kind of similar Bayesian approach. So this is, according to the first passage times, these are kind of the best, the simulated patterns that best recapitulated those patterns observed in experiments. And then these predictions are quite noisy and also model specific, but what they suggested that there was quite a range of different underlying dynamics. But the important message for this work was that we developed this framework to use first passage time to quantify the patterns, and that actually with very little genetic information, you can still extract some important information about the dynamics just by analyzing the geometry of the populations that these dynamics gives rise to. And then last, I'm almost done actually, so I'm just going to quickly motivate what we're working on now in a similar direction, but understanding spatial patterns of extracellular DNA. So ecDNA are basically small fragments of DNA that are broken off into the cell, broken off from the main chromosomal DNA, but persist within the cell. And these can rise to very high copy numbers in the cell, and seem to be, are widely believed to be important in tumorigenesis and resistance, evolution of resistance. Importantly, these lack centromeres and so are segregated randomly during cell division, well described by a kind of binomial partitioning process. But there's much that we still don't understand about observed spatial heterogeneity in ecDNA copy number in tumors. And what we want to do is to understand what mechanisms are driving this heterogeneity and whether we can discern patterns of positive selection, etc. From this. So I'll just quickly flash this slide because I'm out of time, but we have some kind of spatial data where you can see very heterogeneous populations on the left here of ecDNA copy number. And so we're now beginning to use spatial modeling to try and understand kind of what processes drive these observed heterogeneity. But that's me out of time. So thank you all for listening. And if you want to learn more about the first two parts, the liver dynamics and the first passage time, you can find them at these links here. Thanks for listening.
Hi, nice talk. I was just interested in, so that the first passage time metric is an interesting process. I was just wondering whether you compared it to like K nearest neighbor, like distances, like whether you found it was like either computationally more efficient or like just a better metric than kind of the ones out there or not?
Yeah. So actually, in the original paper for the experiments method, they did a bit of modeling and used a spatial Shannon's entropy metric, which was okay, but there were a few sort of important flaws with that. One being that it was only defined for a specific spatial scale, because to compute that entropy, you have to kind of define a grid with a specific grid size that you have to choose. And within those grid sizes, you lose any kind of spatial resolution essentially. So that was one flaw, and that of course is important because these patterns might have sort of correlations over different length scales, and also you can't control all the aspects of the tissue acquisition process as well. So you need a method that is kind of over multiple spatial scales, and so that was one of the benefits that first passage time brought.
Just very quickly, I love this stuff. So you had this one slide where you showed the comparison between the agent-based models and the tumor slice for the genotyping. I don't know if you want to scoot back to that.
Yeah.
So because one thing that the yellow, red, yeah. So the second the second part of the subclonal dynamics part. Yeah, this one.
Yeah,
right?
yeah.
So one of the things that struck me here is that you have this really nice spatial segregation of the individual sectors in the experimental data,
Yeah.
which is obviously a hallmark of these surface expansion models. And I was wondering if that's always the case, or if you also see like fragmented clones where you have intermixing of the two genotypes, which is a bit harder to capture with these types of models.
Yeah, it is. Yeah, we did see that. I mean, we did see. I mean, for example, you see in the top, you see some kind of segregated patterns, and that is maybe typical of what we might see. We wouldn't see maybe anything quite any more dispersed than that. But we did see evidence for that. I would say there was maybe a trend, at least in our model, for predicting sort of more surface-driven dynamics versus like purely kind of exponential growth.
Yeah, for sure. And I think that that's also, that's a great start. But I think many people like, Jake and Maxi and people are realizing, oh, we probably have to look at local frequencies more, right? To actually infer the selection that is happening at that specific clone site. And I think it would be cool to integrate that in these types of models as well.
Yeah.
And thanks a lot. This is amazing.
Thank you.
All right. Thank you very much.